right, let's get started. So I'm super thrilled for us to introduce Erik Hanko. So Erik is our seminar series speaker today. He's also one of the organizers of the Biosensor seminar series. So Erik earned both his bachelor's and his master's degree in biotechnology from the Technical University of Braunschweig in Germany, and then completed his PhD in molecular microbiology at the University of Nottingham. His doctoral research there focused on identifying and characterizing biosensors for various platform chemicals, and then applying those in the chemolitho autotroph Cupria vitis nicator to enable production of platform chemicals from carbon dioxide. Since 2020, Erik's been a postdoctoral researcher at the Manchester Institute of Biotechnology, where he works on the metabolic engineering of model and non-model organisms for the biosynthesis of fine chemicals. His research interests include natural product biosynthesis, transcription factor-based biosensor mining, and their application in enzyme evolution and dynamically regulated biosynthetic pathways. So with that, I'd like to introduce Erik to the seminar series.
Thank you, Khaled. Right, let me try to share my actual slides. Okay, so should be seeing my slides now. Perfect. Okay, so as Khaled mentioned, one of the areas that I'm interested in is the mining of transcription factor-based biosensors from bacterial genomes, and this is going to be my talk about a computational tool that we developed about 3 years ago. So just to very quickly recap, what are transcription factor-based biosensors? So they are regulatory proteins that control gene expression in response to a variety of chemical and of the physical signals. And I'm particularly interested in metabolite signals, so primary metabolites and secondary metabolites. Now, transcription factor-based biosensors can be used in various applications. So they can be used in strain engineering, they can be used in diagnostics for detection and monitoring of compounds, for example, in environmental or medical samples. And one area that I'm particularly interested in is to use them as genetic switches in dynamically controlled metabolic pathways. So they can be used as these type of genetic switches in order to balance gene expression and hopefully further balance gene expression in these heterologous pathways that we often implement in other microorganisms to produce various fine and specialty chemicals. Now, it's relatively easy to perhaps balance the expression of simple biosynthetic pathways just by using constitutive promoters. But the larger these metabolic pathways get, the more difficult it is to fine-tune them by using constitutive promoters. So it's actually quite nice to use genetic switches in order to balance expression. Now, if you have a target molecule and you're interested in finding a biosensor for a molecule of interest, there are a range of different resources out there. For example, this publication from 2018 provides a list of range of different small ligands and their associated regulators. A more recent online database is GrooveDB, which I highly recommend having a look at. And I think I checked yesterday, there's about 300 unique ligands associated to transcription factors currently in this database. So if you have a molecule of interest, but it can't be found in any of these databases, what do you do? So again, there's a number of different tools available that you could do to identify a biosensor for a compound of interest. SenseBio is a great tool which utilizes a structural similarity in order to find a biosensor for your compound of interest. So what it does through structural similarity of your input molecule, if it goes through a database, compares it to a list of the compounds which have transcription regulators associated to them. So once you have some sort of interaction established between a ligand of interest and a biosensor, you can perform biosensor engineering in order to improve specificity and sensitivity, for example. Another great tool is SensiPath, which helps you to link a compound, your input molecule that might not have a regulator associated to it, to a compound that has a regulator associated to it, and then provides you with an additional pathway that needs to be implemented in order to detect your undetectable compound. So now all of these tools are still quite, or like utilizing them is still quite time consuming and labor intensive. So I was thinking about what could we do in order to speed up the process of identifying biosensors for compounds of interest. So one other way would be to actually mine them from bacterial genomes. So this is still a project from my PhD where I was working with cumulative trophic Cuprovidis nicata, and what we quickly realized was that Cuprovidis was able to metabolize a range of different aromatic compounds because it lives in the soil. So when we had a look at these all these aromatic compound degrading pathways, frequently what we saw is that there are regulators associated with the compound degrading pathways. And this is basically the idea that we utilize for the biosensor mining tool that we came up with. So what we want to do is we want to utilize a target input molecule, then we want to link it to a pathway that is involved in its degradation. And then through using this heuristic rule-based model of gene organization, we link it to transcription regulators that are in close genetic proximity to these degrading pathways, and just assume that they're involved in their regulation. So what features should a tool like, we called it TFB Miner, have? Ideally, you want to use a specific target molecule as an input. And here we use the KEGG database to perform all of our analysis. KEGG is quite useful because it has various sub-databases where it links different compounds, reactions, genes, and enzymes. So it's very easy to link the different compounds of your analysis. Another thing that we want to do is we want to be able to screen multiple genomes at once. So there's currently about over 6,000 genomes annotated on KEGG, and we want to screen all of them automatically at the same time. And thirdly, what we want is obviously an output from the tool that is easy to understand. So for example, we want to know which organism the predicted biosensor is from, which genes are set to be in its operon, because once we know the operon, we might be able to know its promoter, so that we also know the operator that the regulator potentially interacts with. Then we want to know the regulator ID, and we want to know kind of a scoring system in order to distinguish sensors that are more likely to interact with your compound of interest compared to sensors that might not be able to interact or less likely to be interacting with your compound of interest. So how does TFB Miner, our biosensor mining tool, work? So first of all, you enter in the script, all you have to do is you enter the KEGG ID of your compound of interest. And then what the script does, what the tool does, it then will use your compound as a substrate, and then it records all enzymatic reactions that are involved in its metabolism. So it records the enzymes or enzymatic reactions involved, and that records all the products. So now in the next stage, what it does, it then uses your first product as a substrate again, and then again it records all the enzymatic reactions that are involved in metabolizing your substrate. Again, it records the products, and it does that until it reaches a predefined chain length. So in this case, our chain length was set to 3 enzymatic reactions. Once this is achieved, and then all the enzymatic reactions are recorded, what the tool then does, it then decides whether all these enzymatic reactions, because you have the reactions, you can link it to enzymes. If you have the enzymes, you can link it to genes, and then you also download with the tool together a genome table, an annotated feature table, which then links all the different reactions to genes, and then the tool decides whether all these genes are in close genetic proximity to each other. So it decides whether they could be arranged in an operon. And then the last thing it does, then the tool screens all these different genetic arrangements, whether there's a regulator nearby. So it does this simply by utilizing the annotation feature of this table, and simply by searching words like regulator, repressor, activator to retrieve these functions. And then the last thing it does, it generates a score. So in case in the top case here, 0 would be the perfect score. It means that you have that it was able to identify a regulator that is in direct opposite orientation of your compound metabolizing gene cluster, and then it subtracts points for deviations from the model. Now, we validated the tool utilizing a commonly used inducible system. So in this case, we use the commonly used arabinose inducible system. So it is composed of the transcription regulator, RSC, which controls expression of the arabinose utilization operon in E. coli. So this is how the script looks like. All you have to do is you enter the KEGG ID, and then sometimes you can also optionally specify the length of enzymatic reactions. So in this case, we can say 3. And then what it then does, it then retrieves all the different chains of enzymatic reactions that it can find or retrieve from the KEGG database. So in case of arabinose, it was able to identify 3 enzymatic chains to degrade arabinose into these 2 compounds. Then it also tells you how many potential biosensors were identified. And for each enzymatic chain, it generates a separate output file. So this is how one of the output files looks like. So this is for the output file that contains all the enzymes of the arabinose utilization operon illustrated here. So in the first column, you see the organism code. Then it also tells you the different genes encoding for the different enzymes that are involved in this operon. It gives you the potential operon, then the regulator, the score. In this case, it gave us, again, a perfect score of 0, and then the regulator annotation. And we can see that in case of the arabinose utilization operon, it worked. So the top hit here was the arabinose operon from E. coli with the RSC regulator. Now, the regulator annotation was actually quite a nice addition to the tool, because now you can also see that because we have different families of transcription regulators, commonly we use RSC regulators to control expression of the arabinose inducible promoter, but you can also see that I was able to identify other families of transcription factors which might have different kinetic and dynamic dose responses compared to RSC. It's nice to have other choices for engineering these circuits. Now to benchmark the tool just to see does it actually return biosensors from the literature. I tested 31 different compounds that have regulators known to interact with these compounds. If you do the maths, it will add up to more than 31, but that's just because some of the compounds have multiple regulators annotated with it. So what you can see here is that for the 31 compounds that I tested, in 16 cases, it was able to identify the original biosensor. Apparently, it was also unable to identify biosensors for quite a few of these compounds. This is because either the KEGG database is incomplete, so sometimes the substrates are actually listed as products, or species are not even listed on KEGG. Sometimes the ligand or the ligand resembles the original ligand that is associated with the sensor. Sometimes the regulation is unrelated to ligand metabolism. So, for example, for compounds that activate a transporter, or sometimes the gene organization just simply not match the required model. So, for example, if the regulator protein is encoded within the same operon as the compound metabolizing genes. Now, you don't have to just trust my word here. So this is from a paper that Simon published. So he built a tool that is very similar to TFB Miner, which utilizes a different database. And he performed a similar benchmarking using 100 known ligand biosensor pairs. And what he found is that for 14 of these, they actually overlap. But then for some of the compounds, they're more likely to be found, for example, in the database that he used, and some of them are more likely to be found in the database that I use. So both tools are complement each other quite well, I would say. So now we wanted to just test whether we can utilize the tool to identify a biosensor for a compound that we're interested in. So at the time when we built the tool, I was part of this large consortium where we were interested in compounds derived from the Shigi-mate pathway, and one of the target compounds was mandelic acid. So mandelic acid is a popular building block in the pharmaceutical industry, both enantiomers of this compound, but it's also used as a urinary biomarker, which indicates exposure to ethylbenzene and styrene. Now, I ran the pipeline using both enantiomers of mandalate. So one of them is R-mandalate, for which it was able to obtain some enzymatic change, but then it had some issues again with identifying which one is the product and which one is the substrate. So it didn't actually return any biosensors for the R enantiomer, but for the S enantiomer, it returned a couple of enzymatic chains, and one of the enzymatic chains is able to degrade S-mandalate into benzoate. So when we look at the table that is generated by the tool, we can see that it was able to identify 13 biosensors with a perfect score of 0, and all 13 biosensors are based on a transcription regulator from the LysR type family. So because I didn't want to test or build 13 sensors and test them, the first thing we did is just to do a bit of checking the homology of these different LysR type transcription regulators. We clustered them into 3 different groups. And then I synthesized these 3 regulators and built a wholesale biosensor composed of a sensing module, which is the regulator expressed by a constitutive promoter, and a reporter module, which is the output promoter linked to a fluorescent reporter. Then I put everything into E. coli and supplemented E. coli with both enantiomers of mandalate, mandelamide, and phenylglyoxylate, which are structurally very similar. So we just wanted to see if there's any cross interaction. And then from the 3 systems, and one of them worked quite nicely, we could see that this system from P. hospita was able to interact with the S-mandalate, with the S-enantiomer of mandalate, and with phenylglyoxylate. Now I recorded the dose response, and the last thing that we wanted to try is can we use this biosensor to detect different levels of mandalate in samples. So for example, it could be used to detect mandalate in urinary samples. So but we already had an in-house where various like strains, different strains that produced mandellate at different levels. So we grew these 5 different strains, and then we just quantified mandellate and phenylglycolates using just UPLC. And then we utilized the supernatant of these cultures, added those supernatants to our wholesale biosensor, and we could see that the output from the biosensor correlated well with the asmandellate concentration as quantified with UPLC. Yeah, that is the tool. The paper, you can read up on it. So the paper is way more detailed than what I told you here. The tool is available on GitHub. I'd like to thank a bunch of people, the people who paid for it, the European Union, and especially shout out to Tarek, which is a very talented master student who helped me with the script. And with that, I'm happy to take any questions.
Awesome. Thank you so much, Erik. And to the audience, feel free to put your questions in the Q&A. I'll kick things off by asking a question about sort of the algorithm that TFB Miner uses. You sort of described this process where you're looking for regulators that are linked to metabolic pathways, but those regulators being in sort of close genetic proximity, right? Typically in upstream and in the opposite orientation to the operon. Can you explain a little bit more about what what that typically looks like? Like, what does close genetic proximity look like? Is it 200 base pairs? Is it 1,000 base pairs? And then I'm also really curious about how often or not that rule is broken, right? Either, it's in the same orientation or it's, somewhere else just really distant in the genome.
Yeah. So to answer your first question, so what we use is we use the indexing of genes in order to determine whether the regulator is directly located upstream of the cluster. So we don't actually utilize a defined length on the chromosome. It's rather just the indexing of the gene. So this is something that would be downloaded together with the script as a feature table, which contains all of this information. To answer your second question, so from just looking at the benchmarking, so obviously there are a range of different sensors that have just different architectures. Or different like inducible systems. So the regulator might be part of the operon, as you said. The regulator might be somewhere else completely on the chromosome. What we're hoping to, so I don't have a specific number how often this is the case. What we're hoping to do with the tool is to cover a large enough number of organisms that perhaps in one organism it has evolved that the regulator has the specific arrangement. So just by screening such a large number by chance, we just hope to find biosensors that way. It'll be very hard to predict sensor interaction if the sensor is located somewhere completely different.
Cool. And it looks like we've got a follow-up question here about whether or not if this architecture of a regulator divergently expressed from a gene encoding an enzyme that metabolizes it in eukaryotes. So I think the question is, do we see sort of these same rules in eukaryotes?
I do not know, is the answer. So this is particularly, so the tool is for prokaryotes. So the genomes that we downloaded and that we used to generate the feature table, they're all prokaryotes from the KEGG database. We didn't look at eukaryotes because, well, regulation might be more challenging than in prokaryotes. Yeah.
Great. I'm curious about you sort of hinted at the fact that TFB Miner is still being developed, right? And that you sort of have ideas for things that you want to do. Can you tell me a little bit more about, what are the plans for TFB Miner? Are there, specific features or capabilities that you'd like to build in a future iteration?
hmm So one thing that I always wanted to do is to like Simon's done for Ligify, just have like an online version of the tool, which is a bit less clunky, a bit easier to use, perhaps. Um, Perhaps combining different databases just to cover a larger space. Another thing that I was interested in was to make it a little bit easier to understand the output. The output is so it's quite hard sometimes to pick from the list of candidates that are generated. So sometimes it might be easier to just provide a more limited number of, for example, the of homologs that are quite similar. So just to reduce redundancy in the data set. And then another thing might be to just expand on the different types of genetic arrangements that we're looking at. So for example, a regulator could be part of the operon, which at the moment, the script completely just ignores. So it only takes regulators that are in opposite orientation. Anything else is currently dismissed. So it would be nice to just, fine tune the tool to accommodate all these different things.
Do you
Um,
know the discrepancy between, sort of the results with Ligify versus TFB Miner? Is that reflective of just, the different databases that the respective tools are drawing from, or is there any other sort of observation that you've been able to make that explains the discrepancy there?
I didn't really have a look into which compounds or if there's any similarities between or differences within the compounds that are returned using Ligify and TFB Miner. Um, so I don't know. It could just be the database that is used. Yeah, so integrating both databases might be like putting it together, might be a way forward. Um, did I see
Great.
that
I
Maggie had
think,
a question?
yeah, it's in the chat. So she was asking if you can clarify how to use the tool. And then in addition to the chemical of interest, do you also specify the species of interest when running the tool?
So the tool itself is a script that works in Python. On GitHub, you can download the script, then the feature table that is needed where it gets all the information from. This was about 2 years ago, so we should probably update that at one point. And then there's also an instruction manual how to use the script. And then the second question was, in addition to chemical of interest, you also specify the species of interest when running a tool. You don't specify the organism. It just tries to retrieve biosensors for all different all different genomes from the KEGG database that it can find, but you can easily search. So once you know what organism you're looking for, you can simply search for the organism identifier in the output file. And then if you run the tool against closely or distantly related species, how similar or dissimilar are the outputs? So for example, if we look at, I think we were just looking at E. coli here. So for example, for the arabinose utilization operon, all the top candidates are species of E. coli, different strains. Um,
Great. Thanks, Eric.